Welcome back to Cancer is a Mitochondrial Metabolic Disease Basics. In the last video on anaerobic metabolism or anaerobic glycolysis or energy without oxygen, it seems that I may have muddied the waters and made things less clear than more clear. So I wanted to take today to try to quickly attempt to redeem myself here and make this more clear. So without further ado, let's get into it. All right, as the slide is entitled, what is anaerobic glycolysis? Attempt number 2. So we have gone over glycolysis as a whole, and we've looked at it from a perspective before as both using oxygen and without oxygen. And the last video was about without oxygen or anaerobic conditions, right? So if there's oxygen and there's intact mitochondria and glucose is the substrate we're talking about for energy, it's going to go through glycolysis from glucose to pyruvate, and then pyruvate will get into the mitochondria as this little circle with the arrow shows, and then it'll be used with oxygen and this intact mitochondria under aerobic conditions to make energy. The vast majority of our energy comes from this, 90+%. So what we're contrasting with the last video is the same cell, same intact mitochondria, no oxygen or insufficient oxygen, and now we're needing to make energy quickly and without oxygen. So because there's no oxygen, pyruvate is not going to get into the mitochondria. Pyruvate is going to be shunted away towards lactic acid or lactate, and it's going to be under conditions where there's a lack of or a decreased amount of oxygen, so oxphos cannot happen. So this is called anaerobic conditions. Same process, it's just diverting from aerobic metabolism to anaerobic or without oxygen metabolism. And in particular, we're looking at this reaction at the very end of glycolysis. Here's glucose, here's all the steps, here's pyruvate, the end product of glycolysis, and that pyruvate is not going to make acetyl-CoA in the TCA cycle. It's going to make lactate. Okay, it's going from pyruvate to lactate through an enzyme called lactate dehydrogenase or LDH. Now, the assumptions that I made is that I assumed that you knew, and some of you may know this, is that in biology, it was kind of assumed that it was either one or the other. Either you were making energy through oxidative phosphorylation and all of the pyruvate was going to make oxidative metabolism, or it was going to be lactate formed in the absence of oxygen or functional mitochondria, and there was no gray area. And that has been debunked. There is a gray area where both aerobic metabolism is happening, aka energy is made with oxygen, but lactate is also made, but it's in vastly different quantities. So probably the best example of the metabolic flexibility that's achieved by the human body is muscle cells under different types of exercise intensity. So for example, low intensity, mostly fat, triglycerides, some glucose. It's not all black and white. There's some glucose used, right? And then as intensity goes up, which means that you're requiring more energy than your body can bring in oxygen because you're sprinting or you're doing a fan bike or you're doing something very intense, then you'll see a ramp up of glycogen, which is stored muscle glucose, and glucose, because now we're mostly using, or we're more likely to use a lot more of this anaerobic metabolism of glucose making lactate. At rest, when there's lactate made, the 50% is being eaten up by either the actual tissue itself that's making the lactate or the surrounding tissue. And then if we increase the intensity and we increase the demand on the system and there's more lactate made, then that will be brought back to the liver to be reconverted through gluconeogenesis. Here, pyruvate is being reconverted to glucose through this process called gluconeogenesis in the liver. And then again, this is an energy dependent process. This is kind of like one of the mechanisms behind cancer-related cachexia. I talk about that in the cachexia video. This is called the Cori cycle. The other thing that was maybe slightly confusing was this graphic here. So basically, I'm going to try to explain this the best way I can. So glucose, if it's the primary fuel, is going through glycolysis. We're making pyruvate. Pyruvate is going to get into the mitochondria. Pyruvate is going to go and get oxidized through oxidative phosphorylation. However, lactate is still being made, but under aerobic conditions, that lactate is either going to be consumed by the mitochondria in the same cell or in the local environment. But what does that require? It requires intact, functional mitochondria, and it requires oxygen. So if you're making a bunch of lactate because you're working hard, really hard, and you're making a bunch of lactate through anaerobic metabolism, then that cannot be oxidized here. Or in the case of cancer, we're not talking about cancer right now, but we're talking about normal cells, specifically muscle cells under duress or under exercise conditions, right? So if there's a lack of oxygen in that tissue, then that lactate cannot be oxidized. How is the lactic acid or the lactate reoxidized? All right, let's orient ourselves on this graphic. This is outside the mitochondria, and this is inside the mitochondria. The same enzymes are present both outside the mitochondria and inside the mitochondria. The enzyme is called lactate dehydrogenase. It is a two-way street, even though the arrow is only going one way because we're talking about the utilization of lactate. But it's a two-way enzyme, okay? So it can convert lactate to pyruvate or pyruvate back to lactate. Same thing here. So under aerobic conditions, there is some lactate made, right? 10-to-1 lactate to pyruvate at resting conditions. That lactate is going to be consumed by the same cell that it produced it under aerobic conditions. So basically the lactate can get reconverted back to pyruvate, or it can go inside the mitochondria, get reconverted back to pyruvate. But pyruvate is the step that is required for it to be oxidized by TCA and by the electron transport chain. If there is no intact mitochondria or functional mitochondria, or the oxygen is insufficient, this will not happen. I hope that clears it up. So let's go back to where we started. So this is aerobic metabolism. OXPHOS is being used with oxygen. Glycolysis will convert to pyruvate. Pyruvate will get into the TCA cycle. It will go through the electron transport chain, and with oxygen will make energy through OXPHOS. However, under conditions where the oxygen levels are low, or you have outworked the amount of oxygen present, there is going to be energy made without oxygen. Pyruvate will get converted to lactate under anaerobic conditions, and that lactate will be transported likely back to the liver and or other tissues that have oxygen, where it can be reconverted back to pyruvate and oxidized in those tissues that have oxygen if that's present. I hope that clears it up. If there's further questions, please leave them in the comments. If you like videos like this, please like, share, and subscribe. Until next time.